Hi everybody, and welcome to the micro e-learning program from the Global Parkinson's Genetics program and the Aligning Science Across Parkinson's ASAP. My name is Lim Kai-shi and I'm a trainee at the University Malaya. Today I'm going to talk about LRRK2 as a genetic risk factor for Parkinson's disease. To give you an overview, we will first provide a brief introduction to the LRRK2 genes, its corresponding protein and its normal physiological function. Then we will touch base on the association between LRRK2 and Parkinson's disease and its risk variant. Finally, we will focus on the development of PD therapy targeting LRRK2 and ended with key take-home messages. LRRK2 stands for leucine-rich repeat kinase 2 and is a gene located in chromosome 12. It comprises 51 exons with a total size of 144 kilobases. This gene encodes for a large protein called LRRK2, also known as dardarin, with a size of 286 kilodalton. The LRRK2 protein is expressed throughout the body, with the highest levels found in the kidney, lung, and immune cells, and a modest expression observed in the brain. In the brain, LRRK2 expression is the highest in the putamen, which is the target of a substantia nigra neurons, although it is also measurable in the substantia nigra itself. The LRRK2 protein consists of 7 domains which can be classified into 2 parts. The first part is the enzymatic core, which consists of the rock core tandem with GTPase activity and a kinase domain. The other part consists of armadillo repeat, enchirine repeat, leucine-rich repeat, and a C-terminal of WD40 domain, which are involved in mediating protein-protein interaction and regulating the LRRK2 catalytic core. Collectively, these domains allow LRRK2 to participate in multiple cellular functions, including vesicular trafficking, autophagy, neurotransmission, mitochondrial function, and cytoskeleton dynamics. Consequently, mutations in this gene may disrupt any of these cellular pathways. The link between LRRK2 and Parkinson's disease was initially discovered in 2002 within the Japanese Sagamihara kindred, pinpointing it as a pathogenic factor in Parkinson's disease. Subsequently, in 2004, the groundbreaking work conducted by the group led by Andy and Thomas identified and verified the disease-causing mutation within this gene, ultimately leading to their recognition and recipient of the Breakthrough Prize in 2024. Since then, more than 100 missense mutations in this gene have been identified, but currently at the time of this recording, only 7 have been confirmed to be pathogenic, all of which are located on the enzymatic core. However, the frequency of LRRK2 mutations varies across different populations. For example, the most common causal variant G2019S has the highest prevalence in the North African population and among the Europeans, but it is almost absent in the African and the Asian population. Furthermore, the penetrance of the mutation also varies across age groups. For example, G2019 carriers have a 28% chance of developing PD at the age of 59 and a 74% chance at the age of 79. Other than the causal mutation mentioned before, there are 2 lines of evidence showing that the LRRK2 locus also contains risk-modifying variants. The first line of evidence comes from the study within the Asian populations, indicating that 2 protein coding variants, G2385R and R1628P, increase the risk of PD by approximately 2-fold. These mutations are present at a frequency of approximately 6% in cases and approximately half of that in the control. Few protective variants such as N551K and R1398H, which reduce the risk of PD, have also been identified in Caucasian, Arab Berber, and also the Asian population. The second line of evidence comes from the genome-wide association studies, GWAS, which shown that non-coding variants within the LRRK2 locus may alter the risk of PD. As more evidence indicates an increase in LRRK2 activity as a cause of PD in many LRRK2 mutant carriers, the development of targeted therapy has focused on this gene. There are 2 main strategies involved. First is the inhibition of LRRK2 enzymatic activity, and second is to downgrade the entire gene. To learn more about the current effort in drug development, please watch Module 12 from the Parkinson Disease Genetics for Non-Geneticist course. Despite having a promising therapeutic target, the scientific community faces several challenges. First, since LRRK2 is expressed in multiple parts of the body, inhibiting its activity may lead to side effects. Secondly, the highly variable penetrance of the mutation makes any assumption about the rate of conversion to the clinical phase problematic. Now we have come to the end of this module. I'd like to end it with a few key messages. First, LRRK2 is a large gene encoding a protein involved in various cellular functions. The mutation in this gene will affect the protein expression and lead to the pathogenesis of Parkinson's disease. Current therapeutic targets on LRRK2 are focusing on inhibiting LRRK2 protein enzymatic activity and downregulating the gene itself. Since the disease-causing and risk LRRK2 mutations are ancestry-specific, global collaborative effort in discovery of ancestry-specific LRRK2 mutation is crucial to uncover the full spectrum of LRRK2 variants across diverse populations and to tailor effective diagnostic and therapeutic strategies accordingly. Thank you for listening. I hope you enjoyed the module.